Okay, then let's start with chapter 10, that is biotechnology and its applications. Chapter 10, biotechnology and its applications. Biotechnology, the topics are biotechnological applications in agriculture, biotechnological applications in medicine, transgenic animals, ethical issues. These are the four main topics in this chapter. Now let's start with it. Biotechnology, as you would have learned from the previous chapter, essentially deals with the previous chapter is processes and uses, deals with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants, and animals. The applications of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. Okay, biotechnology, as we have learned from the previous chapter, deals with industrial scale of biopharmaceuticals and biological use of genetically modified microbes. Medicine, we use it to produce medicine, and we use it for genetically modified microbes to get desired proteins as such, or even for other things, fungi, plants, and animals. The applications of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and even energy production. The three critical research areas are providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. Okay, first critical research areas is providing the best catalyst in the form of improved organism. Usually it's a microbe or a pure enzyme catalyst for catalyzing any kind of reaction. Second, creating optimal conditions through the engineering for a catalyst to react. And creating optimal conditions through engineering for a catalyst to act. Research area of biotechnology is best catalyst, microenzyme, creating optimal conditions for this catalyst enzyme to act, and downstream processing technologies to purify the protein organic compound. The organic compound or protein formed is the product, and to purify this is called as a downstream, and various other process under downstream processing. Let us now learn downstream processing technologies to purify protein organic compound. Three critical research areas: providing a catalyst, a good catalyst, the best catalyst, usually a microbe or a pure enzyme. Next, creating a product, for and making this catalyst to work and act, creating optimal conditions through engineering for a catalyst to act. Next, downstream processing, that is purifying the product formed, testing, etc. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. First main topic: biotechnological applications in agriculture. Let us take a look at the three options that can be thought for increasing food production. Okay, three options that can be thought for increasing food production. What are the things we can do to increase food production? First one, agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture. What is this agrochemical based agriculture? Pesticides, growth regulators. This leads to high input farming, rapid increase in yield, short-term benefits, long-term concerns, soil and water pollution, health hazard. Okay. We can use agrochemical based agriculture for increasing production. Organic agriculture we can use, and genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, making it three times, but yet it was not enough to feed the growing human population. Increased yields have partly been due to use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemical fertilizers and pesticides. Okay, it is a fact that green revolution succeeded in tripling, meaning it it made the food supply three times its normal one. But still, because India is a very large country, it's still not enough to feed the growing human population. The increased yields we have is mainly due to improved crop varieties, like the crop may be resistant to bacteria, pathogens, insecticides, insects, etc. But mainly it is only a small percentage is due to improved crop varieties. It is mainly due to why the increased yield is mainly due to better management practices, better management practices and use of agrochemicals. However, for farmers in the developing world, agrochemicals are often too expensive, and further increases in yield with existing varieties are not possible using conventional breeding. Okay, as traditional breeding techniques fail to keep pace with demand and to provide sufficiently fast and efficient systems for crop improvement, another technology called tissue culture got developed. What does tissue culture mean? It was learned by scientists during 1950s that whole plants could be regenerated from explants, that is, any part of a plant taken out and grown in a test tube under sterile conditions in special nutrient media. This capacity to generate a whole new plant from any cell explant is called totipotency. You will learn how to accomplish this in higher classes. It is important to stress here that the nutrient medium must provide a carbon source such as sucrose and also inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. By application of these methods, it is possible to achieve propagation of a large number of plants in a very short duration. This method of producing thousands of plants through tissue culture is called as micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown, that is, they are soma clones. Many important food plants like tomato, banana, apple, etc., have been produced on commercial scale using this method. Try to visit a tissue culture laboratory with your teacher. Okay. What they're saying is, although green revolution has increased the yield, it is mainly due to uh, management practices and use of agrochemicals. But because the farmers of the developing world are not able to afford agrochemicals, the further increase in yield, how can normal farmers who cannot afford agrochemicals afford to increase their yield? They can't afford agrochemicals. Then they can't get good varieties out of conventional breeding of their available varieties. That's not possible. That's not going to keep up with the efficiency of our needed yield. So Our traditional breeding techniques failed to keep up pace with demand and to provide sufficiently fast and efficient. Okay, then, as traditional breeding techniques failed to keep pace with demand and to prove sufficiently fast and efficient system to crop management, there was a new technology introduced. This was called as tissue culture. This was learned by scientists during the 1950s that when you take a vegetative part of a plant known as explants, any part of a plant taken out and it is grown in a test tube under sterile condition in special nutrient media, this explant will grow into a whole new plant. The capacity to generate a whole plant from any cell of this plant or an explant is called as totipotency. This is important to remember. You will learn how to accomplish. Okay, it is important to stress here that the nutrient media, we need special nutrient media to for these explants to grow into new plants, and the nutrient media must provide a carbon source such as sucrose. It also should have inorganic salts, vitamins, amino acids, and growth regulators like auxin, cytokinins, etc. Cytokinins, etc. Now, by applications of these methods, it is possible to achieve propagation of large number of plants in very short durations. This method of producing thousands of plants, okay, wait. By application of this method, it's possible to achieve propagation of large number of plants in very short durations. Okay, the application of this totipotency method, vegetative reproduction method, basically, we are able to attain thousands of plants in very short durations. This is called as micropropagation. Each of these plants will be genetically identical because we are taking the same cells of a plant. There is no sexual reproduction or anything like that. Because of that, completely identical genetic material. So from one superior variety of a plant, we will get thousands of superior varieties of the plant from that single cell of that plant. Each of these plants will be genetically identical to the original plant from which they were grown. They are called as soma clones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. Okay. Another important application of this method, that is tissue culture, is the recovery of healthy plants from diseased plants. Even if the plant is infected with the virus, the meristem, apical and axillary, is pheomeristem. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plants. Scientists have succeeded in culturing meristems of banana, sugarcane, potato, etc. Okay, one more important application of this method is the recovery of healthy plants from diseased plant. Even if we have a very superior variety diseased plant, like this is not talking about scientists, about normal farmers, they can't just get superior variety of plants for free. So if they have the varieties and they were diseased by virus or something like that, they can isolate the meristem of the plant, that can be apical meristem or axillary meristem, which is free of virus. They can isolate it and get new plants from that meristem, and it will be virus free plants. Hence, one can remove the meristem, grow it in vitro to obtain virus free plants. Scientists have succeeded in culturing meristems of banana, sugarcane, potato, etc. Scientists have even isolated single cells from plants, and after digesting their cell walls, have been able to isolate naked protoplasts surrounded by plasma membrane. and digested their cell walls have been isolated naked protoplasts surrounded by plasma membranes, isolated protoplasts from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoblasts, which can further grown to form a new plant. Okay, scientists have isolated single cells from plants, and after digesting the cell wall of this single cell, they were able to isolate the protoplast, the plasma membrane surrounding the cytoplasm. These isolated protoplasts from two different varieties of plants, each having a okay, this might be referring to two different varieties of the same plant. Two different varieties of the same plant. After the protoplast are fused, we will get a hybrid protoplast, and each have a desirable character. From two different varieties have a desirable protoplast can get fused to get a hybrid protoplast, which can be further grown to form a new plant. These kind of hybrids are called as somatic hybrids, while the process is called as somatic hybridization. Somatic hybridization, where you isolate single cell from different varieties of plants, digest the cell wall, and fuse the protoplast to get a new hybrid protoplast. This is called as somatic hybridization. And the somatic hybrids form the somatic hybrids. Imagine a situation when a protoplast of a tomato is fused with that of a potato, and then they are grown to form new hybrid plants combining tomato and potato characteristics. Well, this has also been achieved, very wild to think, resulting in formation of a pomato. Unfortunately, this plant did not have all the desired combinations of characteristics for its commercial utilization. Although this we are breeding things like this, it is still uncontrollable. Just like desirable characters are getting multiplied and included, even undesirable characters are getting included and multiplied, because we are not specifically taking a single wanted gene and incorporating into the new plant. We are mixing the whole genome. Okay. The plant did not have all the desired combinations of characteristics for its commercial utilization. Is there any alternative path that our understanding of genetics can show so that farmers may obtain maximum yield from their fields? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment are reduced? Use of genetically modified crops is a possible solution to the above questions. Plants, bacteria, fungi, and animals whose genes have been altered by manipulations are called as genetically modified organisms. Important to remember, GMO, plants, bacteria, fungi, or whatever they might be, organisms which have been whose genes have been altered by manipulation or genetic alteration are called as genetically modified organisms, represented by GMO. GM, genetically modified plants, have been useful in many ways. Genetic modification has made crops more tolerant to abiotic stresses, cold, drought, salt, etc. Abiotic meaning non-living, or natural stresses or natural problems that can be cold, drought, salt, heat, reduced reliance on chemical pesticides, pest resistant crops. We can get pest resistant crops. We can make crops tolerate abiotic stress by genetical modification, help to reduce post harvest losses, increased efficiency of mineral usage by plants. This prevents early exhaustion of fertility of the soil. Enhanced nutritional value of food. Example, golden rice, that is vitamin A enriched rice. Important to remember. Important to remember that is golden rice has vitamin A enriched rice. In addition to these uses, GM has been, that is genetic modification has been used to create tailor-made plants, specifically made plants to supply alternate resources to industries in the form of starches, fuels, and pharmaceuticals. Some of the applications of biotechnology in agriculture that you will study in detail are the production of pest-resistant plants, which could decrease the amount of pesticide used. Bt toxin is produced by a bacterium called Bacillus thuringiensis, Bt for short. Bt toxin, Bacillus thuringiensis toxin, gene has been cloned from bacteria and then been exposed in plants to provide resistance to insects without the need for insecticides. Effect created a biopesticide. Example are Bt cotton, Bt corn, rice, tomato, potato, and soybean, etc. Basically, there is a Bt thuringiensis, a bacteria, and this bacteria produces toxin called as Bt toxin. And the gene that produces toxin is been isolated from that bacteria and has been incorporated into the plants. So the plants will also produce Bt toxin, and this will become a biopesticides. This Bt toxin will only harm insects. It cannot harm animals. That means humans. Because of that, the insects will die due to the Bt toxin, but it will act as a natural insecticide or a bioinsecticide. And examples for the Bt plants, the Bt toxin producing plants are cotton, corn, rice, tomato, potato, and soybean that have been made till now. Bt cotton, some strains of Bacillus thuringiensis produce proteins that kill certain insects such as Lepidopterans, tobacco budworm, armyworm, Colaptorans, beetles, and dipterans, flies, mosquitoes. Dipteran meaning two wings. Bacillus thuringiensis forms protein crystals during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Why does this toxin not kill the bacillus? Actually, the Bt toxin protein exists as inactive protoxins, but once an insect digests the inactive toxin, it is converted into an active form of toxin due to alkaline pH of gut, which solubilize the crystals. The activated toxin binds to the surface of midgut epithelial cells and creates spores that causes cell swelling and lysis and eventually cause death of the insect. Okay, this some strains of bacillus, some types of bacteria, some strains of bacteria, this Bt bacteria produce proteins that kill certain insects such as tobacco budworm, armyworm, beetles, dipterans, flies, and mosquitoes. This this Bt bacteria forms protein crystal during a particular phase of their growth. We are talking about the toxin which is produced at a particular time of their growth that form crystals, protein crystals. These crystals contain a toxic insecticidal protein. This is a specific insecticidal protein. Why does this toxin not kill the bacillus bacteria? Actually, this Bt toxin protein exists as an inactive protoxins. It's in an inactive state before entering the insect gut. This is an inactive protein before. But when the insect injects the inactive toxin, it is converted in an active form due to the alkaline basic pH of the gut, which will solubilize, which will dissolve the crystals. Then the active toxin, which will become active from inactive state, will bind to the surface of the midgut epithelial cells and create pores that cause cell swelling and lysis, breaking of cells, and eventually cause the death of the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. The choice of genes depend upon the crop and the target pests, as the choice of genes depends upon the, like, there are many kinds of genes that can create the toxin. The choice of genes depend upon this, the, like, which kind of gene we want will depend on the crop and the targeted pest which will target the plant or which we want to kill. As most Bt toxins are insect group specific, the toxin is coded by a gene CRYIAC named CRY. There are a number of them. This toxin is actually coded by a gene named as CRYIAC or CRYLAC. For example, CRYLAC, it's I here, I believe, not an L, but it is easier to remember as CRYLAC. And short cry. There are a number of them, for example, number of genes. The proteins encoded by the genes CRYLAC and CRYLAB, basically it is CRY1AC and CRY2AB, control the cotton bollworms. That of CRYLAB controls corn borer. Okay. The proteins encoded by the genes, the CRY1 gene and CRY2 gene, CRY1AC and CRY2AB. These CRY1AC and CRY2AB control cotton bollworms. CRY1AB, which will control corn borer. These three types of genes, where CRY1AC will control, along with CRY2AB will control cotton bollworms, and CRY1AB will control the corn borer. These are plant photo given here, where A is destroyed by bollworms and B, it's a fluffy mature cotton ball. Both are cotton plants, but one is destroyed by bollworms and the other is a fully mature cotton ball where you get cotton from. Pest resistant plants. Several nematodes parasite a wide variety of plant and animals, including human beings. A nematode, Meloidigyne incognitia, infects the roots of tobacco plants and causes a great reduction in yield. Meloidogyne, Meloidogyne incognita, Meloidogyne incognita. Okay. So a nematode, it is Meloidogyne incognita, infects the root of tobacco plant and causes a great reduction in yield. Okay, there's a nematode incognita, infects the root of tobacco plants and causes a great reduction in yield. A novel strategy was adopted To prevent this infestation, which was based on the process of RNA interference, RNAi. RNAi or RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing a specific mRNA due to complementary double-stranded RNA molecule that binds to and prevents translation of mRNA. Silencing. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements, transposons, and replicate via an RNA intermediate. Okay, this is whatever is given here is very important to remember. Okay, a novel strategy for adopted to prevent the interference. I mean, prevent the infestation of this nematode, Meloidogeny incognita, that infects the roots of a tobacco plant and causes great reduction in yield. The strategy was that what was RNA interference. This RNA interference is a very important line here. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. It is a method of cellular defense in all eukaryotic organisms. This method involves silencing of a specific mRNA. mRNA is messenger RNA due to complementary double-stranded mRNA that binds to and prevents translation of mRNA. The source of this complementary RNA could be from an infection by viruses, genomes, or mobile genetic elements. I see that. Okay, RNA interference is a cellular defense method in all eukaryotic organisms, and this method involves silencing a specific mRNA. Let's see that. Let's read this as well before. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells, the two RNAs being complementary to each other from a double-stranded RNA and initiated RNA interference and thus silencing a specific mRNA of the nematode not survive in a transgenic host expressing specific interfering RNA. Transgenic plant therefore got itself protected from the parasite. Okay, what we are doing here is we are using agrobacterium vectors. Nematode specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA from a double-stranded dsRNA. In the host cells, they formed antisense and sense RNA. Okay, these two RNAs they are complementary to each other, and they formed a double-stranded RNA that initiated the RNAi, that is RNA interference, and thus silenced the specific mRNA of the nematode. The consequences was that problem is a plant parasite named whatever the name is infects tobacco plant roots. Formation of root galls and causes reduction in the yield. This used RNA interference to protect the plant. What is RNA interference? It's cellular defense found in all eukaryotes. In RNA interference, a specific mRNA, which normally makes a protein, which is normally getting translated into a protein, is silenced. How? When a double-stranded RNA is present, it binds complementary mRNA of the organism. This prevents translation. No protein is made. The target organism gene is silenced. So RNAi basically switches off genes using double-stranded RNA. But where does this double-stranded RNA come from? I'm not sure. It's they mentioned something. It might be coming from infection by viruses having RNA genome or mobile genetic elements. So RNA interference basically switches off genes using dsRNA, that is double-stranded RNA. They used agrobacterium vectors, the same ones used in making transgenic plants. In tobacco plants, they introduced DNA sequences from the nematodes. Into the tobacco plants, they use DNA sequences from the Okay, then that was an interesting method. Let's continue. Pest resistant plants, several nematodes. Parasites have a wide variety of plants and animals, including human beings. A nematode, I need to pronounce that name again. Meladogeny incognita. Meladogeny incognita infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on processes of RNA interference. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing a specific mRNA due to complementary double-stranded RNA that binds to and prevents translation of the mRNA. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements that are jumping genes transposons that replicate via an RNA intermediate. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host. These two RNAs, being complementary to each other, formed a double-stranded RNA that initiated RNAi and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interference RNA. The transgenic plant therefore got itself protected from the parasite. These are the gals. Infects the roots of the tobacco plants. Host plant generated double-stranded RNA triggers protection against nematode infestation. Roots of a typical control plant's transgenic plant root 5 days after a deliberate infection of nematode but protected through novel mechanism. Okay, this is infestation without, this is not modified plant. A is not modified plants, has this kind of formation here, root infestation, and here it is also infected, but this is a modified plant infected by nematodes, but there is no formation of such things because all the nematodes died. Biotechnological application in medicine, the next main topic. The recombinant DNA technological processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Okay, further, the recombinant therapeutics do not induce unwanted immunological responses as it is in common in case of similar products isolated from non-human sources. At present, about 30% recombinant therapeutics have approved for human use the world over. In India, 12 of these are presently being marketed. This recombinant medicine does not induce unwanted immunological responses. As like before these kind of things were there, we used to, in case insulin, it was generated from other animal sources and inputted into humans because if insulin was not there, like before this method was invented, but that caused a lot of unwanted immunological responses. But there were no such things present in the recombinants. do not induce unwanted immunological responses. At present, about 30 recombinants have been approved for human use all over the world, and in India, 12 of them are being marketed. Genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at regular time intervals. What would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate insulin from other animals that would cause immunological responses here. Would the insulin isolated from other animals be just as effective as that secreted by the human body itself, and would it not elicit an immense immune response in the human body? Now imagine if the bacteria were available that could make human insulin. Suddenly, the whole process becomes so simple. You can easily grow a large quantity of bacteria and make as much insulin as you need. Think about whether insulin can be orally administered to diabetic people or not. Why? Insulin used for diabetics were earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source, though caused some patients to develop allergy or other types of reactions to the foreign protein, insulin consists of two short polypeptide chains, chain A and chain B, that are linked by disulfide bridges. Chain A and chain B linked by disulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone, like proenzyme. The prohormone also needs to be processed before it becomes a fully mature and functional hormone, which contains an extra stretch called as a C-peptide. This C-peptide is not present in the mature insulin and is removed during the maturation into insulin. A main challenge for production of insulin is using rDNA techniques was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. Chains A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. Okay, let's read that again. This line is basically okay. Other animals, effective now imagine bacteria available. Insulin-used diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked by disulfide bridges. This is chain A, blue color is chain A, B is chain B, gray is chain C. All the three chains are polypeptides and they are linked by disulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone also needs to be processed before it becomes fully mature and functional hormone. And this consists of an extra stretch called a C peptide. The C peptide is not present in a mature insulin and is removed during maturation into insulin. The main challenge for producing insulin using rDNA techniques was getting insulin assembled into a mature form. We can easily produce the chains, that is A, B and A, B chains, but the problem was assembling it into its like mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences, one DNA sequence for A chain and one for B chain, and introduced them into plasmids of E. coli to produce insulin chains. Chains A and B were produced separately, extracted and combined by creating they were created separately, they were produced separately, A and B peptides, and they are combined by creating disulfide bonds to form human insulin. Maturation of proinsulin into insulin. Simplify. Now let's go to gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. A hereditary disease, a corrective therapy for such a disease. That is the gene therapy. Gene therapy is a collection of methods that allow correction of a gene defect that has been diagnosed in a child embryo. Here, genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of normal gene into the individual or embryo to take over the function of and compensate for a non-functional gene. That allow correction of a gene defect that has been diagnosed in a okay. Here, genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene. Okay. Gene defects normally happen due to absence or misfunction of a specific gene. To correct this, gene therapy has a collection of techniques. The technique is to implant cells having the correct gene that is going to replace the misfunctional gene and produce a specific protein that is required. Inject them into the tissues of the person cells or the embryo. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase ADA deficiency. The first clinical gene therapy was given in 1990 to a 4-year-old girl that is having a deficiency, that is having a gene defect of adenosine deaminase ADA deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But the problem with both these approaches is that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusions of such genetically engineered lymphocytes. However, if the gene isolate from marrow cells, producing ADA is introduced into the cells at early embryonic stages, it could be a permanent cure. Okay, it's still not a permanent cure. The main idea is that if there is a dysfunctional gene that is not producing a protein, uh, a new gene that is replacing this old functional gene, old misfunctional or dysfunctional gene, a new gene is added into the cells of the patient, into the tissues and cells of the patient so that this new gene will work instead of the old dysfunctional gene. That is the main idea. And the first clinical therapy was given in 1990 to a 4-year-old girl. It was adenosine deaminase ADA deficiency. This is a enzyme that is crucial for immune system to function. It's very important. Why this disorder is caused is due to a deletion of a gene for adenosine deaminase. Adenosine deaminase is an enzyme. It is produced from a gene. A gene needs to synthesize this enzyme through transcription and translation. But because of this lack of a gene, this disorder is caused and ADA deficiency is there. In some children, ADA deficiency can be cured by bone marrow transplantation when bone marrow is transplanted, or by directly taking injection. We have, just like when there is a deficiency of insulin, people take injections of insulin. Here, ADA deficiency, people take injections of ADA, the enzyme ADA. Or a first method is bone marrow transplantation, when we transplant the bone marrow or we take ADA injections. These both are a temporary cure to the ADA deficiency problem. But the problem with both of these approaches is that they are not completely curative. And the first step towards the another third method is gene therapy. This is also not a complete cure if it is done at the if if it is done at later stages. It can only be a complete cure if it is done at early embryonic stages. That is when the fetus is still in the womb. What the gene therapy does is, first, lymphocytes are uh, isolated from the patient's body. They are grown in a culture outside the body. Lymphocytes, basically white blood cells, are grown outside the body of the patient. Then a ADA producing gene is incorporated into a into these lymphocytes using a retroviral vector, basically a plasmid. They use genetic modification, and they use recombinant DNA, and they will induce, like, they will incorporate it into the lymphocytes. These lymphocytes, which are grown outside, are again subsequently returned to the patient. These genetically modified lymphocytes are returned to the patients. Because these cells will also die, although these cells have the gene that produces the ADA, it will only be for some time, till the life cycle of the lymphocytes. It will not be forever. However, if the gene isolate from the marrow cells producing ADA, the marrow cells are the ones produce ADA. If these are introduced into cells at early embryonic stage, it could be a permanent cure. Now let's go to the next topic, that is molecular diagnosis. You know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Very conventional methods of diagnosis are serum and urine analysis. Early detection is not possible. Recombinant DNA technology, polymerase chain reaction, PCR, and enzyme linked immunosorbent assay (ELISA) are some of the techniques that serve the purpose of early diagnosis. Okay, first information is that we know that diagnosis is very important for treating a disease. Diagnosis and early diagnosis and understanding the disease is very important for effective treatment of the disease. But using conventional methods, that is serum and urine analysis, early detection of some diseases is not possible. But with recombinant DNA technology, polymerase chain reaction (PCR), and enzyme-linked immunosorbent assay, that is ELISA test, are some of the techniques that serve the purpose of early diagnosis. Presence of a pathogen, bacteria, virus, etc., is normally suspected only when the pathogen has produced a disease symptom. Yes, by this time, however, the concentration of pathogen is already very high in the body. However, very low concentration of a bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. Can you explain how PCR can detect very low amounts of DNA? PCR is now routinely used to detect HIV in suspected AIDS patients. It is used to detect mutations in genes in suspected cancer patients. It is a powerful technique to identify many other genetic disorders. How we can identify is because of the amplification, because it PCR can make the DNA billions of times copies, billions of times of amplification. We can easily detect the HIV or cancer in the patients. We can also identify many other genetic disorders. A single gene strand, a single stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photograph film because the probe will not have complementary with the mutated gene. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins, etc., or by detecting the antibodies synthesized against the pathogen. Okay, there seems to be a second method here. If a single DNA or RNA, it is having a radioactive molecule, which we use as a probe, is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. That took a little longer than expected. Okay, then transgenic animals, the next main topic. Animals that have had their, wait a second, I think we did not complete this. ELISA is based on principle of antigen-antibody interaction. Infection by the pathogen can be detected by the presence of antigens, proteins, glycoproteins, etc., or by detecting the antibody synthesized against the pathogen. By detecting if there are any antibodies produced in the body against the pathogen, we can find out if the person is diseased or not. This is ELISA is a test used in finding out AIDS, so it's very useful. Transgenic animals. Animals that have their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, fish have been produced. Although over 95% of all existing transgenic animals are mice, why are these animals being produced? How can man benefit from such modifications? Okay, what are transgenic animals? Transgenic animals are animals who have their DNA manipulated to possess and ex- express an gene, a foreign new gene are known as transgenic animals. Many kinds of animals have been modified to become transgenic animals, but most of them, 95% of them, are all mice. Let us try to explore some of the reasons as to why these animals are being produced and how can man benefit from such modifications. First one, normal physiological and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. Examples, study of complex factors involved in growth, such as insulin-like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects that result, information is obtained about the biological role of the factor in the body. Okay, we this is for study. Study is how genes are regulated and how they affect normal functions of the body and its development. Study of complex factors involved in growth, such as insulin-like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects that result, information is obtained about the biological role of factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specifically made to serve as models for human diseases so that investigation of new treatment for diseases is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease. Third reason, biological products. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of portion of DNA which codes for a particular product, such as a human protein, alpha 1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria and cystic fibrosis. In 1997, the first transgenic cow, Rosie, produced human protein enriched milk, 2.4 grams per liter. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Well, let's see the above reason and this one again. study of disease, many transgenic animals are designed to increase our understanding of how genes contribute to development of the disease. These are specifically made to serve as models for human diseases where investigations of treatment for diseases is made possible. The transgenic models exist for many human diseases such as cancer, cystic fibrosis. Okay, biological products, medicines required to treat certain human diseases contain biological products. These are expensive. Transgenic animals that produce useful biological products can be created by introduction of the portion of DNA which codes for particular product such as human protein alpha 1 antitrypsin used to treat emphysema. Similar attempts are made for treatment of phenylketonuria and cystic fibrosis. Transgenic cow rosy human enriched milk. The milk contained human alpha-lactalbumin, which was nutritionally more balanced product for human babies than natural cow milk. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccine before they are used on humans. Transgenic mice are being used to test the safety of polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safeties of batches of the vaccine. Chemical safety and testing. This is known as toxicity safety testing. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain results in less time. And ethical issues, the next main topic, the fourth last topic. The manipulation of living organisms by the human race cannot go on any further. Without regulation, some ethical standards are required to evaluate the morality of all human activities that might help or harm living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organizations such as GEAC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of genetic modification research and the safety of introducing GM organisms for public services. The modification usage of living organisms for public services as food and medicine sources, for example, has also created problems with patents granted for the same. There is growing public anger that certain companies are being granted patents for products and technologies that make use of genetic material, plants, and other biological resources that have long been identified, developed, and used by farmers and indigenous people of a specific region slash country. Rice is an important food grain. The presence which goes back to thousands of years in Asia's agricultural history. There are estimated 200,000 varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati rice are grown in India. There is reference to basmati in ancient texts, folklore, and poetry as it has been grown for centuries. In 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed company to sell a new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from India's farmers' varieties. Indian basmati was crossed with the semi-dwarf varieties and claimed as an invention of a novelty. The patent extends to functional equivalents, implying that other people selling basmati rice could be restricted by the patent. Several attempts have been also made to patent uses, products, and process based on Indian traditional herbal medicine, example turmeric, neem. If we are not vigilant and we do not immediately counter these patent applications, other countries, individuals, many may encash on our rich legacy and we may not be able to do anything about it. Biopiracy is the term used to refer to the use of bioresources by multinational companies, other organizations without proper authorization from the countries and people concerned without compensatory payment. Biopiracy is a term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure during their commercialization. There has been growing realization of the injustice, inadequate compensation, and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patent Bill that takes such issues into consideration, including patent terms of emergency provisions, research and development initiatives. Summary: Biotechnology has given to humans several useful products by using microbes, plants, animals, and their metabolic machinery. Techniques of tissue culture and somatic hybridization offer vast potential for manipulation of plants in vitro to produce new varieties. Recombinant DNA technology has made it possible to engineer microbes, plants, and animals such that they have novel capabilities. Genetically modified organisms have been created by using methods other than natural methods to transfer one or more genes from one organism to another, generally using techniques such as recombinant DNA technology. GM plants, that is genetically modified plants, have been useful in increasing crop yields, reduce post-harvest losses, and make crops more tolerant of stresses. There are several GM crops with improved nutritional value of foods and reduce the reliance on chemical pesticides. Recombinant DNA technological processes have made immense impact in the area of healthcare by enabling the mass production of safe and more effective therapeutics. Since the recombinant therapeutics are identical to human proteins, they do not induce unwanted immunological response in case of similar product isolated from non-human sources. Human insulin is made in bacteria, yet its structure is absolutely identical to that of the natural molecule produced in humans. Transgenic animals are also used to understand how genes contribute to development of a disease by serving as models for human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease. Gene therapy is the insertion of genes into an individual's cells and tissues to treat diseases, especially hereditary diseases. It does so by replacing a defective mutant allele with a fundamental one or gene targeting which involves gene amplification, virus that attack their host, and introduce their genetic material into the host cell as part of their replication cycle are used as vectors to transfer healthy genes or more recently portions of genes. The current interest in manipulation of microbes, plants, animals has raised serious ethical questions, which you have seen. With that, first revision of this chapter completed,